Hey everybody, this is Daylon and Arun, hosts of the STEMCELL podcast. We're back on day 3 of our video series, rounding up the latest research presented at the 2020 ISSCR virtual meeting. In addition to these videos, don't forget to follow us on Twitter at STEMCELL Podcast to stay on top of everything we're doing at the meeting, including a fun little game in which you can try to match an ISSCR speaker with a quote pulled from their STEMCELL podcast episode. Be sure to check it out. We've got some interesting research from the meeting that we want to highlight today, but before we get to that, as every day, Arun has a brief message for you. Arun.
I do. Watch the Innovation Showcase Advanced Brain Organoid Co-Culture Systems from Stem Cell Technologies for an overview of the development and applications of complex HPSC-derived 3D neural models. The live presentation and Q&A will happen tomorrow at 1 p.m. Eastern Time. Find the full schedule for live sessions at STEMCELL's virtual booth.
All right, Arun, we left off yesterday in the afternoon. It was right before the presidential plenary, the first one. I was excited to get that, but I was not so excited to be a part of it. Why don't you give us your impression?
Yeah, I thought it was well done given some of the technical issues that we had. But on that note, I mean, even in person, you have technical issues, right? PowerPoints crash, videos don't work. So kudos to ISSCR President Deepak Srivastava for facilitating things and helping things move along. I was actually looking at the chat as well. And I think people in general were really receptive and positive. They just accepted that this happens. It's inevitable. This is part of the modern reality. I remember Deepak was filling in some of the time by telling a couple of stem cell jokes that theoretically went well, according to people in the chat. So one of them was like, what was it? What did the one stem cell say to the other? Let's split and get out of here. I mean, yeah, sure. Okay, well.
Yes, there are dad jokes and then there are stem cell jokes. I don't know if Deepak's a dad, but he might have doubled down right there. The technical glitches notwithstanding, I thought it was like you said, and the chat, I think, kind of agreed. Once you were able to get over the technical obstacles, I thought it was a solid group of presentations. I really enjoyed David Scadden leading off. He had a couple nice little elements that hadn't occurred to me. One was that the whole HSCI, no, not the HSCI, that Moderna was started by a seed grant from the HSCI, which I thought was a really cool thing. And the other thing, just in terms of the timing, was that, and you just realize listening to these guys that, they've been there at the beginning. And right now we're watching these young investigators who are at the beginning of their career. And it really struck home when Dr. Scadden talked about the previous epidemic that brought him into the fold, which was the HIV epidemic, decades ago. And now here we are again with COVID. So who knows whose careers are starting in the midst of these tumultuous times. But we know that Dr. Scadden kind of was invented in those times. And then also there was the Public Service Award, which really was touching to me. I was one of the first NICEF postdoctoral fellows, the Druckenmiller fellows. So I've known Susan Solomon for a long time, and I have a lot to thank her for. And also there was a really nice tribute video from some of the luminaries and stem cells that you guys can check out on ISSCR. So that was really touching. And she said a few words also, although we couldn't see her. She was really had a lot to say that was really both touching and also really insightful and impactful, and put into fine relief the impact that she's had on the field as a whole. So that was great. And then there was Aviv. Arun, talk to us.
Well, we all know about Aviv Regev, who is the single cell extraordinaire, formerly at the Broad Institute, now has actually moved to take an industry position. Of course, Aviv was Aviv, right? She's reconstructing fate maps using single cell lineage analysis and throwing in some machine learning in there too. So if you had to think about actually a theme that was sort of permeating some of these talks, I actually felt like machine learning and automated analysis of stem cell differentiation, that was something that could have, that was sort of the taking over. Of course, she kicked things off with an incredible talk of doing what she does in all things cell biology. And after that, we had Stephen Finkbeiner from the UCSF, searching for signatures of neurodegenerative and psychiatric diseases. So he was also using iPSCs to model ALS, and he was focusing also on a machine learning kind of application, where they were actually looking at disease phenotypes in their ALS neurons, and they're able to actually identify the populations of cells that had disease phenotypes based on machine learning, just based on neuron morphology in a dish. So that's kind of back to back machine learning applications. Then we had Eric Olson, who's Eric Olson, one of the grandfathers of cardiac basic biology, and he's over there at UT Southwestern, talking about using CRISPR with Duchenne muscular dystrophy. Not really a cardiac specific application per se, but a lot of translational potential there. And then wrapping things up with Fiona Watt, who is an expert on all things skin biology. Her title of her talk was Adventures in Skin Biology, and she was talking about the epidermal compartment and epidermal stem cell differentiation. So we covered the gamut from experts in different topics across the body. Really a great way to, I think, to kick off this meeting.
Yes, unfortunately for any that didn't catch it live, it is now a ghost. You know, they had to take it off, I guess, because it was such a nightmare technically. Arun mentioned in yesterday's video episode that these things are online for 30 days. I'm hoping they can get at least some of that session back on. You know, maybe not as a whole, but some of the parts. I mean, I was howling when Aviv. I mean, a couple of things. Like you said, looking at her presentation, I saw how like there's all these papers that we take for granted now with all of that Seurat plots that are just like it's standard now, and like she invented that. You know, I mean, she and her postdoc who now has his own lab, Satya. But the point being there is that all this that we look at and take for granted as gospel, it all began with just these algorithms. And I was laughing when she said a couple things. One, she was like, "I have 5 vignettes for you. You know, anyone else goes in there, they got 15 minutes, right? And they're like, okay, so I'm going to give you 1 vignette that's representative. Aviv comes with 5 because she's got 30, and I mean, time must stretch out for her. She has so many studies because she referred to a study. She was like, yeah, there was one study that we had several years ago, and that was published in 2018. So, Aviv, you're doing too much, all right? It's a good thing you're taking a break with your industry position over there. You know, you earned it. I hope you enjoy it. And moving into today, we had the second plenary, which was really great. Started off with I should just mention, there was the late night talk that you were at too, Arun. I was at Mark Tomashima led that from Blue Rock Therapeutics. I'm going to haze him a little bit because I went there for the synthetic biology because I'm pretty familiar with the CAR T, although that was pretty amazing too. But Mark, in introducing the synthetic biology component, he emphasized how it could be utilized, and he reduced it down to, I'm not kidding, this is no exaggeration, he reduced it down to flavored beer, flowers that turn colors during the day, barbecue, and leather. Okay, so I mean, we're trying to save the world here, all of us investigators, and Mark Tomashima over there at Blue Rock Therapeutics. While he cares about that, he also cares about, the finer things in life as well. But yeah, I went there for the editing, the more, synthetic biology and logic control gates type stuff on that. I'm not going to get into it. That was very interesting talks from Sean Cordoba and Tim Liu, who were both in private industry. But you were there for the CAR T stuff, right? The Tregs and the macrophage that are in candidacy for therapy. Tell me about that.
Yeah. We're not gonna dive into it too deep, and I don't want to make a fool of myself just because it's not my area of expertise. But yeah, really nice couple of opening talks to that session. First was from Bruce Blazar, looking at Tregs, T regulatory cells, and hopefully using iPSC technology to perhaps derive those cells. One issue that he actually mentioned, and this is sort of obvious, is that if you want to make personalized iPSC derived T regulatory cells, it's expensive, right? Ideally, you would want to have a batch of these cells that can cover the entire population. But if you truly want to make it personalized to to an IPS level, that's going to be a really expensive therapy. So that's one thing that's holding back personalized IPS technologies when it comes to that and the intersection of immunoengineering. So that was one thing. He also introduced base editors and CRISPRs to actually further modify these cells. The second talk was pretty neat. It was from Courtney Crane. She was doing something similar when it came to with the T regulatory cells, but focusing more on the macrophage side of things, and in particular, how these genetically engineered macrophages that she was making, how they can actually target solid tumors pretty well. We have an idea of how these CAR T cells and macrophages can tolerate liquid tumors and are pretty good for treating leukemias and stuff, but solid tumors is the next big thing, and her work was focusing on that. And so that actually brings us to today. So today's second plenary, focusing on machine learning and computational approaches. Like I mentioned, that was sort of following from Aviv Regev and some of the initial themes that we developed during the first presidential plenary. One of the talks that really blew both of us away was Alan Klein. He was the award for the outstanding young investigator, and he's talking about learning dynamics by lineage tracing on transcriptional landscapes. So why don't you give an overview of that one?
Well, this isn't exactly my field either, but like you said, it's the emphasis on machine learning, and it's clear. We think about we talk always about single cell seq, and also organoids, of course. But the single cell seq, that's all at its root based on the technologies that these investigators have developed, Aviv Regev and also Alan Klein. And I think the thing that makes them so amazingly prolific is that they're fundamental, technologies. We're talking like PCR. They're ways of reframing your approach, and so they have these collaborations and everything. And you look, Alan said it. He said all these species he listed in very quick succession. What he didn't mention is that every one of those species was like science, cell, nature, cell biology. All these developmental, fundamental, organisms that are at the pillars of developmental biology. Now we're gleaning new insight. And he went through a number of things that he was doing in the lab, and even mentioned some work they had on in press, this CLINC, cell lineage from normalized covariance. And my takeaway from that really was that I think we're moving into an era where the machine learning, and it's never going to be a perfect substitute for human, but it can complement the human mind in a way that humans aren't capable of thinking in certain ways, in the same way that computers aren't capable of thinking or feeling in certain ways yet. And I think the takeaway here was that he had these approaches and algorithms and all this, this machine learning that's beyond me. But what it did is that it predicted an event that was controversial, the cell lineage within hematopoiesis, and suggested an alternate progenitor for a major cell type, and confirmed it. And then in this news story that's in press, suggested another novel that wasn't so hinted at, but there was support for in the literature. So I think we're getting to this point now, and Alan Klein's and Aviv as well, I think their talks both kind of indicate, and there's a lot of posters and other talks that are hinting at this too, is that we might be leaning on AI as a hypothesis generator a little bit more moving into the future, because we've kind of run out of creative ideas maybe.
Yeah, there was an extreme example of that in the last talk of the session, and I'll get to that in a minute, from Hiroaki Kitano over in Japan. But second talk in the plenary 2 session was from Emma Lundberg over there in Sweden. She's talking about the spatiotemporal dissection of the human proteome. She's one of the leaders when it comes to the human proteome atlas. Really had some beautiful immunofluorescence schematics and images, and seeing how to combine that, basically use those images to look at protein localization in a variety of different cell types. And one thing that she actually mentioned that kind of stuck with me was, even in a theoretically homogeneous population of identical cells, clonal cells, there is significant variation when it comes to the protein localization and protein expression of even a single protein. So, she's showing images of just like 10 cells in a field. And within those 10 cells, the subcellular localization of a certain protein was completely dramatically different. All right. So that tells you that there's still variability in these theoretically homogeneous populations. Another thing that she actually mentioned, which has been published, I think it was published a couple of years ago in Nature Biotech, was how she actually used citizen science to do some of this analysis looking at subcellular protein localization. She actually farmed out some of this analysis to gamers at EVE Online, which is like this massively multiplayer online game. And so there's like a little mini game within the overall game that where the gamers were basically identifying different protein compartments in these cells and got points for it or something like that. So I thought that was a really, really unique application of citizen science and something that honestly I think we should do more of.
Yeah, that was very cool. And I had heard about that a while back, but it was nice having it circle back in the context of stem cells and this human protein atlas. And the stat that jumped out at me, I thought was cool. It was more than 300,000 players
Yeah,
that had engaged with this, and they had contributed in aggregate 70 working years
right.
um, to the project. So I did the math because, I'm a mathematician. That averages 2 hours each, which is significant. It's a
Oh
mini
yeah.
game within a video game. I don't know if you've ever played a video game, but if you're spending 2 hours on it, it's got to be pretty fun. Another new note that I thought was cool because Emma, she was gracious enough to answer and post all the Q&A in the
Mm.
Q&A tab. You saw all the questions and what was posted there. She said that now this has been repurposed. In the past, it was repurposed to identify exoplanets or something, but
Yeah,
now
right.
it's being repurposed to analyze flow plots of COVID samples. So this is something, like you said, we should be doing more of. Well, she beat you to that. This is being repurposed already to fight the current scourge of humankind. So yeah, a really cool application there.
Yeah, so there are a couple of other talks wrapping up that particular session. There's Ajameda Kekas looking at next generation human iPSC models, again, using machine learning approaches to distinguish between disease and control cells and iPS models. One thing that they mentioned was the output is always dependent on how your model is trained. So garbage
Mm.
in, garbage out, right? That's what machine learning is all about. You have to have a good teaching to actually get anything good on the other side. Next up, Trey Edeker from UCSD, also talking about the intersection of machine learning, and this is sort of relevant to what I do, intersecting machine learning to connect autism spectrum disorder to congenital heart disease. And then after identifying some proteins and some pathways that are important in linking both of them, they're actually able to validate what they found through machine learning in a Xenopus model. So bringing it back to the true basic science. And finally, wrapping it up with the talk that I alluded to, Hiroaki Kitano from Japan. He was, I think there's a reason that he was last in this session. He was basically saying, all right, we're going to take this to the next level. We're going to use machine learning to take away all of our jobs. All right. We're going to basically go from the inception of projects all the way to the actual analysis and experimental design. We're going to tell machine learning to do all of that. And is that a realistic possibility within the next few years? And that's kind of what he was discussing. I don't know. That was out there, but hey, maybe it's the next step.
Yes, controversial. I wouldn't go so far. I mean, self-preservation, Arun. I'm trying to live here. He could say he's probably a few days from retirement, but I'm trying to work.
Right.
So yeah, let's get into the concurrence now. The first concurrent of the session, I was kind of anxious about the concurrence because
Mhm.
it's a lot. It's a lot of action, but and the timing really, because sometimes I don't know about you guys, but real concurrence, I find that it never really lines up. No
Right.
matter the best laid plans. So I was anxious, but I thought it worked out pretty well. We both sat in for the first session with Martin Pera to, the first to open the concurrence. We listened to Martin Pera because we're going to have him on the show. And I thought about him that was really refreshing is it really brought me back to like first days in stem cells when they were being derived and everyone was leaning on meth and conditioned media, and they were aggravated, I guess, and they were looking. The big innovation, what everyone was seeking, was a recipe for maintaining pluripotency, and that was it. That was everything. And Dr. Pera is still grinding on that because while we've come up with recipes that work and that are practical and that have really moved the field forward, I don't know that we've really answered the question. So Para was talking about heterogeneity in his human ES cell colonies and grinding with kind of similar approach, similar assay, similar material, but with the real new age technology. And what struck me as like I thought of as I was listening to him is that this is a guy at Jackson Labs. Jackson Labs famous for the mice, but they let him study human embryonic stem cells there. So this is a guy who can do whatever he wants And it shows because he's just doing good work, doing good hard work, basic science with real life implications in stem cell biology.
Yeah, and as I mentioned yesterday, with regard to Sergio Pasca, Dr. Pera is also going to be a guest on our podcast. But I think you knew that one, Daylon,
Yes,
right?
I did know
So,
that. That's in the schedule. But then, yeah, there was also I'm in repro a little bit now, so I got with a lot bit. I love my job in repro, boss, chief. That's for you, chief. But Jennifer Rosenbluth, who is a postdoc, I just want to give her a shout out because she was from Joanne Brugge's lab, but was given the stage and talked about her work with mammary organoids. And the real takeaway for me, which was cool here, is that she took organoids in 2D versus 3D, which is it's a basic idea, but she did it in a really, I think, elegant way, and then took this reductive approach, taking out all the components of the media to see what's really important for growth and the distribution of cells at high level, by the way, using single cell seq, and then kind of applied that to normal or malignant cells. So giving an idea of how we can maybe target these malignant cells in the breast in situ. So I like that from a young investigator who really clearly has a long career ahead of hers. And I think, Arun, then you directly after that, you caught the session after that when I bounced, right?
yeah, absolutely. So there were a ton of great sessions in this. I actually think that the concurrent sessions are really the meat and potatoes, the real heart of the conference, because this is when young PIs and young investigators and trainees really get to get their shine on, right? So they get to present a lot of unpublished work, and that's actually something that I was really excited and happy about, that there was unpublished work that was being presented here. Like, for example, we'll get to June Woo in a little bit later. That was pretty mind blowing. But a couple of talks that I went to included Roseanne Raftery, who's using single cell transcriptomic analysis to evaluate articular cartilage differentiation. Also, Hao Wu, more on the cardiac side of things. I had to go to one cardiac talk. Okay, Daylon, I kept it under 50%. I promise you. So he was focusing on the molecular mechanisms of heart development using all sorts of seq. But I think the emphasis here was he really is focusing on nuclear ATAC and nuclear RNA-seq, which is kind of the next generation of these single cell sequencing technologies. Sylvia Velasco using brain organoids to study development and also identifying mutations that can cause a shift in the population of different cells in the brain organoids. And one of my favorite talks was with from Samira Musa, who's actually a new PI at Duke University. I think formerly she was in Boston. She's actually focusing on kidneys, so iPS-derived kidney tissues and podocytes. Actually, something that doesn't get too much love on this podcast. She's able to combine kidney differentiation with organ chips to actually develop a system that can mimic the filtration of the kidney using these iPS-derived podocytes. So that was really nifty. Another organ chip technology paper or presentation was from Sean Harrison over there in Norway, using tissue scale culture of human PSC-derived liver organoids and high sensitivity analysis techniques. So similar to Dr. Musa, Sean Harrison was using organ chips, but this time in combination with liver organoids, and was actually looking at some drug metabolism using both these technologies. One thing he actually looked at was active heroin metabolism, which I think that's exciting. It's something that you don't really hear about every single day. And then we actually shifted into the animal chimera research section, and I think both of us were on that talk, on those talks. So why don't you wrap that up?
Yeah, there was the high-end, heavy-duty ethics and then Jun Woo component, but leading into that, there was a lesser-known little mini chimera story that I want to highlight from a young investigator, Maureen Krish, who is a postdoc in Yanish lab, and I thought it was really brave of her to show unpublished data where they used fMRS, or they used it Fragile X, induced pluripotent stem cells and then made chimeras in mouse brains and looked at competition between normal and these fMRS mutant neural derivatives. So I thought that was a really cool story, and brave for her to share her data and this unpublished data. I'm sure we'll see it in a few minutes. But getting to the nitty gritty, there was an ethics presentation from Josephine Johnson. I mean, I don't want to go too deep on that because I think it was the same ideas that we covered yesterday about the science and ethics of brain organoids in terms of those ethical conundrums, which really, to me, the takeaway is really about the language. It's about making sure that the public doesn't think that we're making like chimeras that come out of a horror movie. You know, we got to be very clear about what we're doing there. So, I mean, I don't think it was anything that scientists don't know, but other than to say that we need to be more careful in our explanations and stop trying to come up with sexy names. You know, like one thing she said that I thought I was totally behind is the basic term saying humanized. Whenever you say humanize something to a layperson, they're going to assume that whatever that is then more human. Okay, so when you're surprised, then they're throwing up their arms saying, "What the hell?" It's you shouldn't be, you know, because we haven't been very clear. And when you leave the room for that kind of murk, you're gonna get the dregs coming up with bad ideas, and they're gonna use it as ammunition to shut down your science. So I think it's just the takeaway for me there was that you got to be careful, and it was nice because seamlessly you went to the most intense chimera stuff that exists. And Arun, you can segue into that if you like.
Yeah, absolutely. So then we wrap things up with Jun Woo, who's over there at the UT Southwestern in Texas. Cell competition constitutes a barrier for interspecies chimerism. I mean, just to preface this, I'm a big proponent of this work because I actually think this is really the way of the future. And if you want to truly obtain a huge supply of replacement organs, I personally think that this is the way to do it. Back when, you know, Hiromitsu Nakauchi at Stanford demonstrated that you can generate a rodent pancreatic hybrid, I think that kicked off the field. And the dream down the road is to create animals potentially that are harboring these potentially human organs. And one of the researchers who's at the forefront of doing this kind of work is Jun Woo. A lot of what he presented was unpublished work, so props to him. And one thing he actually led off with was the fact that this is so freaking hard to do. The efficiency for these processes and these, even the failure, even just to get some engraftment of some human cells into an early stage non-human embryo is exceptionally difficult. But he was actually focusing more on the in vitro side of things, and he focused on an assay that I think is going to be published pretty soon, looking at cell competition. So basically, to see how different human pluripotent stem cells from different animal populations compete with each other in a dish, and to see which of these cells actually takes over in the culture. So he compared HPSCs from humans, from cows, from rats, from mice, and also from macaques to see how they actually compete with each other in a dish. And he said one thing that actually I thought was really neat was he mentioned that there's like a hierarchy. There's a hierarchy of different HPSCs from different species that actually take over in the culture. So if you introduce a human pluripotent stem cell population with a mouse, then the mouse is gonna take over. And one thing that he also mentioned was that a lot of this competition happens in the context of primed pluripotent stem cells. Of course, we're still working towards the naive state. And then he wrapped up his talk just mentioning, oh, this isn't the only thing that we're doing. We're also looking at these million other things that are preventing, human animal chimerism from happening. So there's so many different evolutionary barriers that you have to think about. But I'm a big fan of this. I really do think it's the way of the future. But of course, you got to do it right. So.
Yes, I mean, wave of the future in terms of endless supply of organs. I think it's going to be hard to get past the yuck factor there, but
Sure.
maybe not. But yeah, in terms of the study, it was just so simple and elegant, just all in vitro. And then trying to use that lever that, I think was a great thing for me to look at what alterations you could make to the cells. In this case, he was knocking out NF-kappa B signaling to make them kind of sneak through that competition and show that it could work in vitro. I know everybody was waiting for them to show, some chimera evidence, but I'm sure it's forthcoming. And those experiments take a long time and are very challenging, as Arun said. But the big thing that I took away is in the post, there was questions, of course, about how long do you let these chimeras go and the ethics and all that. And Jun Woo, I think he had a great answer, which was that we don't have to go into deep, development, fetal stages where you have development of the central nervous system. We don't have to do that right now. To answer the questions we want to answer, we can keep it simple, we can keep it safe, we can keep it on the right side of all the bioethics and all that. So I think he's taking a remarkably careful approach, to his credit, where I think a lot of people are asking this question: show me a heart, show me a pancreas. And he's saying, no, we're trying to get contribution. And as soon as we see that we've surmounted that obstacle, we can terminate that pregnancy, because we're not interested in making those organs now, and especially not interested in doing it in a way that would be controversial or would worse, pose some serious bioethical questions that I just have intrinsic concerns to them. So I love Junwoo. I want to have him back on the show. He was on now almost probably over 2 years ago. So we'll get him back on maybe when this paper comes out. Just a quick note, because, we got to give a little love to the poster sessions out here and the young trainees. There was one about BioSilk in the first session, BioSilk from BioLamina. I think it's going to be really good for 3D. There's this air-liquid interface culture from Christine Abo. Paul McKeever from UToronto, he was looking at organoids and ALS in a way that was super intense and clinically relevant. Kim Leonhard from YCell. I think importantly, we got to give credit to these people. They've been karyotyping. They got 10 years and thousands of karyotypes where they did a comparison between ES and iPS cells, looked at G-band karyotyping across all those to give you just the numbers, and this is what science is about. It's the details and the numbers. And finally, there was this Fraunhofer decellularized ureter thing that I thought was so cool. So if you guys have a minute and you get into the post session, check that out. That is going to do it. But before we go, I just want to make a note. We talked a lot about the immune engineering. This is a note to try and get in there for stem cell technology. You should check out an upcoming product from Stem Cell Technologies, serum-free stem diff immune kits that are used to differentiate human pluripotent stem cells into T cells, NK cells, or monocytes, all very relevant to the immune system. You can sign up now to be the first to know when it's available at www.stemcell.com/ISSCR 2020-FTK. Thank you guys so much for joining us for this third of the video episodes. We're going to be back tomorrow and the next day for a total of 5. We'll hope you'll join us then too.